Right now, as you listen to this, you're not alone. Thousands of creatures are moving across your skin, crawling through the landscape of your eyelashes, burrowing into the valleys between hair follicles. They're hunting, feeding, reproducing. They've been there your entire life, and you've never seen a single one. We think of invasion as something that comes from outside, ships crossing oceans, armies at borders. But the most successful invasions on Earth happened so quietly, so completely, that we didn't even notice. They colonized every human who ever lived. They turned our bodies into continents. These aren't parasites in the way we usually think of them. They're ecosystems, food chains, predators hunting prey across distances measured in micrometers, and the terrain they navigate, it's you. What we're about to discover might change how you think about where you end and the rest of life begins. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture the surface of your skin. You probably think of it as a barrier, a boundary between inside and outside. Smooth, continuous, yours. But that's not what it is. Not really. Zoom in close enough, and your skin becomes something else entirely. Mountains and valleys, caves and forests, rivers of oil flowing between ridges of dead cells stacked like geological layers. And moving through this landscape, invisible to your eyes but absolutely real, are hunters. They have 8 legs, translucent bodies, claws designed for gripping surfaces you can't even feel. Some are elongated, worm-like, living head down in the tunnels of your hair follicles. Others are round, armoured, scavenging through the debris fields of shed skin cells. They're born on you. They die on you. Entire generations pass without ever leaving the few square centimetres they call home. You're a world to them, and they've been living here longer than you have. This isn't science fiction. This is ecology. Right now, on an average human face, there are somewhere between 1 and 2 million of these organisms. They have names: Demodex follicularum, Demodex brevis, dozens of species of mites, springtails and microarthropods that have evolved specifically to live on mammalian skin. They don't live anywhere else. They can't. We are their only habitat. But here's what makes this strange. What turns these creatures from simple parasites into something more complex. They're not just living on you. They're hunting each other. There are predators and prey, grazers and scavengers, species that farm bacteria, species that eat the farmers. Your skin isn't just hosting individual organisms, it's hosting food webs, complete ecosystems, with energy flowing from one species to another, with competition and cooperation, with birth and death and evolution happening at speeds we're only beginning to understand. For most of human history, we had no idea they existed. We couldn't see them. The microscope had to be invented first, then electron microscopy, then genetic sequencing that revealed just how many species were actually there, how diverse these communities really are. Every new tool we developed to look closer showed us more life, more complexity, more strangeness, living in a place we thought we knew completely. And the more we looked, the stranger it got. Because these organisms aren't randomly distributed. They have preferences, territories. Some species live only on faces, others only in ears. Some prefer oily skin, others dry. They've partitioned your body the way species partition a rainforest, each one carving out a niche, a way of surviving that doesn't directly compete with its neighbours. Your eyebrows are a different habitat than your cheeks, your scalp is a different world than your forearms, and the creatures living in each location have adapted over millions of years to thrive exactly there and nowhere else. But the strangeness doesn't stop at your skin. Open your mouth, and there are different species. Hundreds of bacterial species, yes, but also protists, microscopic fungi, organisms feeding on the constant flow of food particles and dead cells. Your gut is even more crowded, trillions of bacteria forming communities so complex that scientists are still mapping them, still discovering what they do, how they talk to each other, how they talk to you. You're not one organism, you're a collection, a walking ecosystem. And most of the cells in that ecosystem aren't even yours. Tonight we're going to trace these invisible invasions from the outside in. from the mites on your skin to the bacteria in your gut, from the ecosystems on your eyelashes to the predators hunting in your bed. We'll discover how these organisms got here, what they're doing, why you need some of them more than you'd ever guess, and what happens when the balance breaks. Because understanding these invasions changes everything. It reveals that the line between self and other, between you and the environment, isn't a line at all. It's a gradient, a negotiation, a conversation that's been happening since the first animals evolved skin, since the first gut learned to digest food, since the first immune system learned to distinguish friend from threat. Let's begin where all invasions begin, at the border between inside and outside. Maybe the invasion started the moment you were born, before that actually. In the womb, you were mostly sterile, protected. The amniotic fluid kept the microbial world at bay, and your skin was yours alone. But the instant you entered the birth canal, everything changed. Your mother's microbiome became yours. Bacteria from her vaginal tract, from her skin, from the air in the delivery room, all of it rushing to colonize the blank slate of your newborn body. Within hours you were covered. Within days, ecosystems were establishing themselves. Species that arrived first claimed the best territory. Species that came later had to find whatever niches remained. And once established, these early colonizers defended their ground, producing chemicals that killed competitors, forming biofilms that were nearly impossible to displace. This is called priority effects in ecology. First arrival matters. The microbiome you developed in your first few days of life shaped the microbiome you have now, decades later. Those early invasions set the terms for every invasion that would follow. But we're not starting with bacteria. We're starting with something bigger. Something you can almost see, if you had the right equipment. Something with legs and eyes and a hunger that's been shaped by millions of years of evolution to exploit one very specific resource: you. Let's talk about the mites, Demodex follicularum. The name means worm of the fat, and it's more literal than you'd think. This creature, roughly 1/5 of a millimetre long, lives in your hair follicles, not on your skin, in it. Head down, body extending up the narrow shaft of the follicle, legs gripping the walls, mouth parts feeding on the sebum, the oily secretion your skin produces constantly. According to research published in 2022 by Alejandra Perotti and her team at the University of Reading, nearly every adult human on Earth hosts these mites. They're not evenly distributed though. They prefer your face, especially the areas around your nose, your eyelashes, your forehead. Places where sebum production is highest, where the food is richest. During the day they stay hidden, buried in the follicle, barely moving. But at night, when you're asleep and your skin temperature drops slightly, they emerge. They crawl across your face at speeds that seem glacial to us, but are sprinting for them. They're searching, for mates mostly, for new follicles to colonize, for territories that haven't been claimed yet. They have no anus. This fact seems trivial until you realize what it means. They can't excrete solid waste. Everything they eat, everything they digest stays inside them until they die. And when they die, which happens after about 2 weeks of life, they decompose. Right there in your follicle, their bodies break down, releasing all the accumulated waste at once. Your immune system notices. Sometimes it overreacts. There's growing evidence that Demodex populations, when they get too large, contribute to rosacea, that chronic redness and inflammation that affects millions of people. The mites aren't the sole cause, but they're a factor, a trigger. Their waste products, released in concentrated bursts when they die, can inflame the surrounding tissue. But here's what's strange. You need them. Not in the way you need your gut bacteria, perhaps, but their presence isn't purely negative. Research from 2024 by Henk Brage's lab at Bangor University suggests that Demodex mites may actually help regulate sebum production. By feeding on excess oils, they prevent pores from becoming completely clogged. They're gardeners, in a sense, pruning back overgrowth, maintaining a balance. The problem isn't their presence, it's their overpopulation. And that overpopulation happens when something else changes, when your immune system weakens, when your skin's pH shifts, when the bacterial communities that normally keep mite numbers in check get disrupted. Because the mites aren't alone in those follicles. Living alongside them, sometimes inside them, are bacteria. Bacillus oleronius is one. It's been found living in the guts of Demodex mites, and when the mites die and decompose, these bacteria are released. and Bacillus oleronius produces proteins that trigger immune responses in humans. Inflammation, redness, the exact symptoms of rosacea. So it's not just the mites, it's the mites plus the bacteria they carry. It's a nested ecosystem, one species hosting another, and your face hosting both. The invasion is layered, each organism enabling others, creating conditions that allow even more species to thrive. And Demodex follicularum isn't even the only mite species on your face. There's Demodex brevis, shorter, rounder, preferring to live in the sebaceous glands themselves rather than the follicle shaft. It feeds more directly on the gland cells, occasionally causing them to swell and malfunction. It's less common than follicularum, but it's there, present on most adults, occupying a slightly different niche in the same general habitat. Two species, closely related, dividing up the resource. This is niche partitioning, one of the fundamental principles of ecology. You see it in rainforests, where dozens of bird species feed on insects, but each specializes in a different height, a different hunting technique, a different size of prey. You see it on coral reefs, where fish species coexist by eating different things, hiding in different crevices, being active at different times of day. And you see it on your face, where two mite species have evolved to exploit human skin without directly competing. But mites are just the beginning. Move beyond your face, down to your arms, your legs, the broader landscape of your skin, and the community changes. Demodex mites thin out. Sebum production drops. The environment becomes drier, less hospitable to oil feeders, and into this space come different organisms. Springtails, for instance, Collembola. These are true insects, though they're primitive ones, lacking wings, usually only a millimetre or two long. They're more common in soil, in leaf litter, in the damp environments where they feed on fungi and decaying organic matter. But some species have adapted to drier conditions, and a very few, can survive on human skin. They're not parasites in the traditional sense, they're not feeding on you. They're feeding on the microscopic debris that accumulates on skin surfaces, dead skin cells mostly. The outermost layer of your epidermis is constantly shedding, sloughing off in flakes too small to see individually, but substantial in aggregate. You lose roughly 30,000 to 40,000 dead skin cells every hour. That's nearly 1 million cells a day. And where there's that much organic debris, there are scavengers. Springtails hop through this landscape, using a specialized appendage called a furcula, a forked structure folded under their abdomen that snaps downward like a spring-loaded catapult. It launches them distances of several body lengths in an instant. For a creature 1 millimetre long, that's a leap of several millimetres. Scale that up to human size, and it would be like jumping 50 feet. They're not hunting, they're grazing, moving from one patch of dead cells to another, feeding, reproducing, living out their entire lives on a surface area roughly the size of a postage stamp. But even these scavengers have predators. Pseudoscorpions, for instance. Tiny arachnids, usually 2 to 3 millimeters long, with pincers like a scorpion but no tail, no stinger. They're ambush predators, hiding in crevices waiting for prey to wander close. And when something does, something small enough to overpower, they strike. Pincers close around the prey, venom is injected through specialized glands, and the victim is slowly consumed. Pseudoscorpions have been found on human skin, though rarely. They're more common in homes and closets in old books where they hunt book lice and dust mites. But occasionally, they venture onto us. Hitchhikers, perhaps. Explorers testing a new environment. Most don't survive long. Human skin doesn't offer enough cover, enough prey density, to sustain a pseudoscorpion population. But the fact that they're there at all, even briefly, reveals something important. Your skin isn't a monoculture. It's not just one species exploiting one resource. It's a patchwork of microhabitats, each supporting different organisms, each connected to the others through invisible threads of predation, competition, and symbiosis. And if we zoom in even further, past the mites and springtails and pseudoscorpions, down to the microscopic scale, the diversity explodes. Bacteria. Tens of thousands of species, most of them unknown to science until very recently. For decades, microbiologists tried to study skin bacteria by culturing them in labs, growing them in petri dishes, identifying them under microscopes. But most bacteria can't be cultured. They require conditions we don't know how to replicate, nutrients we haven't identified, partnerships with other species that we haven't figured out yet. So for a long time, we only knew about the tiny fraction of skin bacteria that could survive on agar plates, maybe 5%, maybe less. Then, in the early 2000s, genetic sequencing technology advanced. Scientists could extract DNA directly from skin samples, sequence it, and identify bacteria by their genetic signatures without ever growing them in a lab. And what they found was staggering. According to the Human Microbiome Project, launched in 2007 by the National Institutes of Health, human skin hosts somewhere between 500 and 1,000 bacterial species, and that's probably an underestimate. Every new study using better sequencing methods finds more diversity, more rare species, more variation between individuals. Your armpit has a different bacterial community than your forearm. Your forearm has a different community than your palm. Your palm has a different community than the space between your fingers. Each site on your body is colonized by species adapted to the specific conditions there. Moisture, temperature, pH, sebum levels, exposure to air, frequency of washing. It's not random. It's structured. Predictable even. Staphylococcus and Corynebacterium species dominate moist areas like armpits. Propionibacterium acnes, despite its name, is actually a beneficial species that dominates sebaceous areas like your face and upper back, feeding on sebum, producing fatty acids that lower skin pH and inhibit the growth of pathogenic bacteria. In dry areas, like your forearms, diversity is highest. There's less competition for resources, more niches available, more opportunities for rare species to establish themselves, and layered on top of the bacteria, occupying different niches, are fungi, Malassezia species mostly. These are yeasts, single-celled fungi that feed on the oils in your skin. They're present on nearly every human, concentrated on the scalp, face, and upper back. In normal populations, they're harmless, possibly beneficial even, competing with more dangerous fungi and bacteria for space and resources. But when they overgrow, which happens when sebum production increases or when the immune system is suppressed, they cause problems. Dandruff, for one. That characteristic flaking of the scalp is often caused by Malassezia feeding too aggressively on scalp oils, triggering an inflammatory response that accelerates skin cell turnover. Seborrheic dermatitis, a more severe condition causing red, scaly patches on the face and chest, is also linked to Malassezia overgrowth. The fungus itself isn't necessarily more abundant in people with the condition, but the immune system's reaction to it is stronger, more inflammatory, creating the symptoms we recognize as disease. So again, it's not about presence or absence, it's about balance, about populations staying within ranges that the immune system can tolerate, that don't trigger chronic inflammation, that don't outcompete the other species that keep the ecosystem stable. And maintaining that balance requires constant negotiation between your immune system and the organisms on your skin. Your skin is not passive. It's not just a surface that organisms happen to land on. It's an active participant in shaping the communities that live there. Your epidermal cells produce antimicrobial peptides, small proteins that kill certain types of bacteria while leaving others unharmed. The specific mix of peptides you produce, which is partially genetic and partially influenced by your health and environment, determines which species can survive on your skin and which can't. You also produce sebum, sweat, dead cells. Each of these secretions contains nutrients, but in different ratios. Sebum is rich in lipids. Sweat contains salts, urea, amino acids. Dead cells offer proteins, keratin, structural molecules. Different organisms specialize in metabolizing different nutrients, so the specific composition of your secretions shapes the community composition, and your immune system is constantly patrolling, identifying organisms, deciding which ones to tolerate and which ones to attack. This process, called immune surveillance, is incredibly sophisticated. It involves pattern recognition receptors that can distinguish between different bacterial cell wall structures, between different fungal components, between species that have been present since birth and species that just arrived yesterday. Most of the time, the immune system leaves resident species alone. It's learned, through constant exposure since infancy, that these species are normal, safe, possibly beneficial. But when a new species arrives, or when a resident species starts behaving abnormally, Multiplying too fast, moving into the wrong tissue layer, the immune system responds. Inflammation is the most obvious response. Redness, swelling, heat. These are signs that immune cells are flooding the area, releasing chemicals that kill invaders but also damage surrounding tissue. It's a trade-off. A controlled burn to prevent a larger fire, but there are subtler responses too. Your skin can adjust its pH, making the environment more acidic, which favours certain bacteria over others. It can alter sebum composition, reducing the availability of nutrients that specific organisms need. It can increase the production of certain antimicrobial peptides while decreasing others, fine-tuning the chemical environment to favour desired species. This is ecological engineering. Your immune system isn't just fighting off invaders, it's cultivating a garden, weeding out species that might cause problems, nurturing species that provide benefits, maintaining a diverse community that's resilient to perturbations. But the garden extends beyond your skin. Open your mouth, and the ecosystem changes completely. Your oral cavity is one of the most densely colonized habitats in the human body. More than 700 bacterial species have been identified in the mouth, and that's probably still an undercount. They live on your teeth, your gums, your tongue, the inside of your cheeks, the roof of your mouth. Each site has its own community, shaped by local conditions. Your teeth, for instance, are unique surfaces, hard, non-shedding, constantly bathed in saliva but also exposed to food, to temperature fluctuations, to the abrasive forces of chewing. Bacteria that colonize teeth have to attach firmly, resist being washed away, and metabolize whatever nutrients happen to flow past. Streptococcus mutans is one of the most famous, though famous for the wrong reasons. It's a primary cause of tooth decay, producing acids as a byproduct of sugar metabolism, acids that dissolve tooth enamel and create cavities. But it's not acting alone. It's part of a biofilm, a structured community of multiple bacterial species encased in a matrix of polysaccharides and proteins. This biofilm, called dental plaque, is incredibly resilient. It resists mechanical removal unless you brush aggressively. It resists chemical attack from saliva's antimicrobial enzymes, and it creates microenvironments within itself, with different oxygen levels, different pH zones, different nutrient availabilities, allowing multiple species to coexist in a space just a few micrometers thick. At the base of the biofilm, closest to the tooth surface, are anaerobic species, bacteria that can't tolerate oxygen. They metabolize nutrients that diffuse down from the surface, producing waste products that feed other species higher up. At the top of the biofilm are aerobic species, bacteria that need oxygen, that are exposed to saliva and food particles, that grow fast and reproduce quickly. It's a layered ecosystem, with energy and nutrients flowing vertically through the structure, with each species occupying a specific niche, with cooperation and competition happening simultaneously. And the biofilm communicates. Bacteria within it produce signaling molecules, small chemicals that diffuse through the matrix and trigger changes in gene expression in neighbouring cells. This is called quorum sensing, a way for bacteria to detect population density, to coordinate behaviour, to switch from individual survival mode to collective action mode. When bacterial populations are low, quorum sensing molecules are dilute, scattered, ineffective. Bacteria operate independently, focusing on growth and reproduction. But when populations are high, when the biofilm is thick and mature, quorum sensing molecules accumulate. They reach threshold concentrations, and bacteria respond by changing their behaviour. Some species start producing more extracellular matrix, thickening the biofilm, making it harder to remove. Others start producing toxins, chemicals that kill host cells or inhibit immune responses. Others become more resistant to antibiotics, activating efflux pumps that expel drugs before they can take effect. The biofilm becomes more than a collection of individual bacteria. It becomes a coordinated system, a collective entity that's harder to kill, harder to displace, harder to control than any individual species would be on its own. And your immune system knows this. It's evolved strategies specifically to deal with biofilms, to break them apart, to penetrate the matrix, to kill the bacteria hiding inside. But it's a constant battle. The biofilm rebuilds, the bacteria adapt, the immune system escalates, and the cycle continues day after day, year after year, as long as you're alive. But the mouth isn't just about teeth. your tongue hosts a different community. The surface of your tongue is rough, covered in papillae, small projections that increase surface area and create countless crevices where bacteria can hide. Anaerobic species thrive here, protected from oxygen, from saliva flow, from mechanical removal. Some of these species produce volatile sulfur compounds, chemicals that smell like rotten eggs or decaying meat. This is the primary cause of bad breath. Not poor hygiene, necessarily, though that makes it worse, but the metabolic activity of bacteria breaking down proteins, producing sulfur as a waste product, releasing it into the air you exhale, and the smell is useful from an ecological perspective. It's a signal. It tells other organisms something about the community composition in your mouth, about what's being metabolized, about whether conditions are favorable or hostile. Predatory bacteria can detect these compounds and move toward them, seeking prey. Immune cells can detect them and increase surveillance in areas where sulfur-producing species are concentrated. Even other humans can detect them, subconsciously, and make judgments about health, about mate quality, about whether someone is worth approaching or avoiding. Breath odor carries information, and that information shapes social behavior, partner selection, even the transmission of oral bacteria between individuals through kissing, shared utensils, close contact. We think of our bodies as private, as individual, as separate from others, but microbially we're constantly exchanging. Every handshake transfers thousands of bacteria. Every shared meal is an opportunity for microbial migration. Every kiss is a mixing event, a chance for oral communities to share species, to acquire new genetic diversity, to test whether organisms from one person's mouth can survive in another's, and some can, some do. There are species that seem to be specialists in human-to-human transmission, that produce adhesins specifically tuned to attach to epithelial cells in multiple individuals, that can tolerate the immune challenges of a new host, that can establish themselves quickly before resident species mount a competitive defense. Other species can't. They're so highly adapted to one individual's specific conditions, one person's pH, one person's antimicrobial peptide profile, that moving to a new host is fatal. They arrive, they try to attach, and they're killed within hours by an unfamiliar immune system, or outcompeted by resident species that are already established, already controlling the best resources. So your oral microbiome is a mix of personal species unique to you and cosmopolitan species, shared across humans, passed from person to person, generation to generation, in an unbroken chain stretching back thousands of years. But the mouth is still just the entrance. The real invasion happens deeper. Swallow, and everything changes. The journey from mouth to stomach takes seconds, but in those seconds, everything you swallowed, food, saliva, bacteria, passes through one of the most hostile transitions in your body. Your esophagus is a tube, roughly 10 inches long, connecting your throat to your stomach. It's muscular, constantly moving, squeezing content downward through waves of peristaltic contraction. And it's relatively inhospitable to microbial life. The walls are smooth, constantly shedding cells, constantly bathed in mucus that makes attachment difficult. There's no steady food supply, no stable environment, just periodic floods of whatever you've swallowed, rushing past, gone in seconds. Most bacteria that enter your esophagus don't survive the journey. They're swept along, carried down into the stomach, where they face something even more lethal: acid. Your stomach is a chemical weapon. Gastric acid, primarily hydrochloric acid, maintains a pH between 1.5 and 3.5. That's strong enough to dissolve metal given enough time, strong enough to break down proteins, to denature enzymes, to rupture bacterial cell membranes on contact. It's a sterilization chamber, designed to kill nearly everything that enters, and for the most part it works. Most bacteria die within minutes of reaching the stomach. Their cell walls break down, their internal machinery stops functioning, they're digested, broken into amino acids and simple sugars, absorbed or passed along as harmless debris. But not all bacteria die. Some have evolved to survive. Helicobacter pylori is the most famous example. This spiral-shaped bacterium lives in the mucus layer coating your stomach walls, protected from direct acid exposure. It produces an enzyme called urease, which breaks down urea, a waste product present in your stomach, into ammonia. And ammonia is a base. It neutralizes acid. So H. pylori creates a bubble of neutral pH around itself, a microenvironment where it can survive, reproduce, and colonize despite being surrounded by one of the harshest chemical environments in nature. According to research published in 2024 by the World Health Organization, roughly half of all humans on Earth carry H. pylori. It's one of the most successful bacterial colonizers of the human body. And it's been with us for at least 100,000 years, possibly longer, spreading through families, passed from mothers to children, evolving alongside us as we migrated out of Africa and populated every continent. For most people, H. pylori causes no symptoms. It just lives there, quietly occupying the stomach lining, feeding on mucus and urea, keeping its population stable. But in some people, it causes problems. It triggers inflammation, weakens the mucus layer, allows acid to reach the stomach wall directly. This leads to ulcers, painful erosions in the stomach lining that can bleed, that can perforate, that can become life-threatening if untreated. And in rare cases, chronic H. pylori infection increases the risk of stomach cancer. The constant inflammation, the constant cellular damage and repair creates opportunities for mutations, for cells to lose their normal growth controls, for tumours to develop. So for decades, the medical consensus was simple. H. pylori is a pathogen. If detected, it should be eliminated with antibiotics. But recent research complicates that picture. Studies from 2023 by Martin Blaser at Rutgers University suggest that H. pylori might actually provide benefits, particularly in childhood. Children who carry H. pylori have lower rates of asthma, lower rates of allergies, lower rates of certain autoimmune conditions. The bacterium seems to modulate the immune system, training it to respond appropriately to threats, preventing it from overreacting to harmless substances like pollen or food proteins. Eliminating H. pylori might reduce ulcer risk, but it might also remove a regulator, a species that's been shaping human immune development for millennia. We're still figuring out the trade-offs, still trying to understand whether H. pylori is a pathogen, a symbiont, or something in between. A species whose effects depend entirely on context, on host genetics, on the presence or absence of other organisms. But whether H. pylori is there or not, the stomach eventually empties. Food, bacteria, digestive enzymes, all of it passes through the pyloric sphincter into the small intestine, and here the invasion reaches its peak. Your small intestine is roughly 20 feet long, coiled and folded inside your abdomen. Its inner surface isn't smooth. It's covered in villi, finger-like projections that increase surface area to about 250 square meters, roughly the size of a tennis court, folded and packed into a space the size of your torso. This is where most nutrient absorption happens. Proteins broken down into amino acids, carbohydrates split into simple sugars, fats emulsified and packaged into micelles, all of it crossing through the intestinal lining into your bloodstream where it's distributed to cells throughout your body. And living on that vast surface, coating every villus, every fold, every crypt, are bacteria. Trillions of them. More bacterial cells in your gut than human cells in your entire body. The most densely colonized habitat on Earth, rivaled only by soil and perhaps the deep ocean floor. The numbers are hard to grasp. Estimates suggest the human colon contains somewhere between 10 trillion and 100 trillion bacterial cells. The uncertainty is massive because counting them directly is nearly impossible, but genetic sequencing gives us a proxy. By extracting DNA from fecal samples and sequencing it, scientists can estimate how many bacterial genomes are present, how many species, how much diversity. And the diversity is staggering. A typical human gut hosts between 500 and 1,000 bacterial species. Some people have more, some have less. But everyone's gut is colonized by a complex structured community that's been shaped by diet, by geography, by genetics, by antibiotic exposure, by birth method, by early childhood experiences. The species present aren't random. They're organized, structured by the environment, by competition, by cooperation, by the constant pressure of your immune system sorting through them, tolerating some, attacking others, shaping the community in ways that promote your survival, because you need these bacteria desperately. Your genome encodes roughly 20,000 genes. The bacterial genomes in your gut collectively encode something like 3 million genes, That's 150 times more genetic information, 150 times more metabolic capability. And much of that capability is dedicated to breaking down molecules you can't digest on your own. Fibre, for instance. Humans don't produce cellulases, the enzymes needed to break down plant cell walls. We eat vegetables, grains, fruits, and most of the structural material in those foods, cellulose, hemicellulose, pectin, passes through our stomach and small intestine completely unchanged. We can't digest it. We can't extract energy from it. It's useless to us, but it's not useless to bacteria. Gut bacteria, particularly species in the phylum Bacteroidetes, produce dozens of different enzymes that can cleave the chemical bonds in plant fibres. They break down cellulose into glucose. They break down hemicellulose into xylose and other simple sugars. They break down pectin into galacturonic acid. And then they ferment these sugars, producing short-chain fatty acids, acetate, propionate, butyrate, as waste products. These fatty acids are absorbed by your intestinal cells and used as energy. Butyrate, in particular, is the primary fuel source for colonocytes, the epithelial cells lining your colon. Without it, these cells starve, the intestinal lining weakens, permeability increases, and you become vulnerable to infections, to inflammation, to diseases like inflammatory bowel disease. So when you eat fibre, you're not really feeding yourself, you're feeding your bacteria. And they in turn feed you. It's a mutualism, a partnership where both parties benefit, where neither can thrive without the other. But the benefits go beyond energy extraction. Gut bacteria produce vitamins. Vitamin K, which you need for blood clotting, is synthesized almost entirely by gut bacteria. Certain B vitamins, biotin, folate, B12, are also produced by bacterial species in your colon. Without them, you'd develop deficiencies, even if your diet was otherwise perfect. Gut bacteria also train your immune system. The largest concentration of immune cells in your body isn't in your bone marrow or your lymph nodes, it's in your gut, in the gut-associated lymphoid tissue, or GALT. Roughly 70% of your immune system is located here, constantly sampling the contents of your intestine, constantly making decisions about what's dangerous and what's not, and it learns by interacting with bacteria. Immune cells, called dendritic cells, extend projections through the intestinal lining, grabbing bits of bacterial cell wall, pulling them back into the tissue, presenting them to other immune cells. This is how your immune system learns to recognize normal beneficial bacteria and distinguish them from pathogens. If you're raised in a sterile environment, if you never encounter diverse bacterial communities in early childhood, your immune system doesn't develop properly. It becomes hyperreactive, treating harmless substances like threats. This is part of the hygiene hypothesis, the idea that reduced microbial exposure in modern sanitized environments contributes to rising rates of allergies, asthma, and autoimmune diseases. According to research from 2024 by researchers at the Max Planck Institute for Evolutionary Biology, children who grow up on farms, exposed to animals and soil and diverse microbial communities, have significantly lower rates of immune disorders than children raised in urban environments. The difference isn't genetic, it's environmental, it's microbial. Your immune system needs to be educated, and the teachers are bacteria. But not all bacteria are teachers. Some are invaders, true pathogens, species that evolved not to cooperate with human hosts, but to exploit them, to extract resources, to reproduce as quickly as possible even if it damages or kills the host. Salmonella enterica is one, Shigella dysenteria is another. These are enteric pathogens, bacteria that invade the intestinal lining, that trigger massive inflammatory responses, that cause diarrhea, vomiting, fever. Your body tries to flush them out to expel them as quickly as possible, and the result is dehydration, malnutrition, sometimes death, particularly in children and the elderly. But even these pathogens aren't invincible. They have to compete with resident bacteria. They have to find space, resources, attachment sites on the intestinal wall. And if your gut microbiome is diverse and stable, if resident species are occupying every available niche, producing antimicrobial compounds, consuming all available nutrients, pathogens struggle to establish themselves. This is called colonization resistance. A healthy microbiome is a barrier, a defense, a first line of protection against incoming threats. it's not perfect. Pathogens can still invade, particularly if they arrive in large numbers, if you're immunocompromised, if you've recently taken antibiotics that wiped out resident species. But a stable community makes invasion much harder, and this is why antibiotic use has consequences that extend far beyond killing the target pathogen. Antibiotics are indiscriminate. They kill susceptible bacteria regardless of whether they're harmful or beneficial. They wipe out entire populations, create ecological vacuums, open up niches that opportunistic species can exploit. Clostridioides difficile, commonly called C. diff, is a perfect example. It's a spore-forming bacterium that's present in low numbers in many people's guts, kept in check by competition from other species. But after antibiotic treatment, when resident bacteria have been killed off, C. diff can bloom. It multiplies rapidly, produces toxins that damage the intestinal lining, causes severe diarrhea and colitis. C. diff infections are notoriously difficult to treat. Standard antibiotics often make things worse, killing more resident bacteria, allowing C. diff to rebound even stronger. The most effective treatment, according to recent clinical trials, isn't more antibiotics, it's fecal microbiota transplantation, or FMT. This is exactly what it sounds like. Stool from a healthy donor is processed, screened for pathogens, and transplanted into the patient's colon, usually via colonoscopy or enema. The goal is to re-establish a diverse bacterial community, to reintroduce the species that were wiped out by antibiotics, to restore colonization resistance. And it works. Success rates for FMT in treating recurrent C. diff infections exceed 90%. It's one of the most effective medical interventions in gastroenterology. And it's teaching us something profound. Sometimes the best way to fight an infection isn't to kill the pathogen directly. It's to restore the ecosystem that keeps the pathogen in check. But the gut isn't just a battleground between good bacteria and bad bacteria. It's a communication network. Bacteria in your gut talk to each other. They talk to your intestinal cells, and increasingly evidence suggests they talk to your brain. This is the gut-brain axis, a bidirectional communication system linking your digestive tract to your central nervous system. It operates through multiple channels: the vagus nerve, which carries signals directly from the gut to the brainstem; immune signaling molecules like cytokines, which circulate through the bloodstream and cross the blood-brain barrier; and microbial metabolites, chemicals produced by gut bacteria that enter circulation and affect neuronal function. Research from 2024 by researchers at the University College Cork in Ireland has shown that gut bacteria can influence behaviour, mood, even cognition. Mice raised in sterile conditions with no gut bacteria at all show altered brain development, increased anxiety, impaired social behaviour. Transplanting bacteria from anxious mice into calm mice transfers the anxiety. The behaviour follows the microbiome, and it's not just mice. Human studies are finding correlations between gut microbiome composition and depression, between microbial diversity and cognitive function, between specific bacterial species and neurological conditions like Parkinson's disease. The mechanisms are still being worked out. But one pathway involves neurotransmitters. Your gut bacteria produce many of the same signalling molecules your neurons use: dopamine, serotonin, GABA. These chemicals are synthesized by bacterial enzymes, released into the gut lumen, and some fraction of them enters circulation, crosses barriers, reaches the brain. Whether they're produced in your gut by bacteria or in your brain by neurons, these molecules have similar effects. They bind to receptors, they alter neuronal firing patterns, they influence mood and motivation and perception. So in a very real sense, your mental state is partly microbial. The organisms in your gut, shaped by what you eat, by antibiotics you've taken, by stress and sleep and exercise, are contributing to how you feel, how you think, how you experience the world. This is still controversial. The field is young. Many studies are correlational, not causal. But the evidence is accumulating, and it's pointing towards something profound. You are not one organism. You're a collective. And the boundaries between self and other, between your cells and their cells, between your metabolism and their metabolism, are far more blurred than we ever imagined. But we're not done descending. The gut is densely colonized. Yes. But it's not the end of the invasion. There are organisms that go even deeper. Beyond the gut, Past the intestinal lining where trillions of bacteria live in negotiated peace, there are places we once thought were sterile. Sealed chambers, sacred spaces where blood flows clean and organs perform their work undisturbed by microbial life. We were wrong. For over a century, medical science operated on a fundamental assumption. Healthy internal tissues are sterile. Blood is sterile, the bladder is sterile, the uterus is sterile. The lungs, except for transient microbes breathed in and quickly expelled, are sterile. Disease happens when these barriers are breached, when bacteria invade from outside, when sterility is compromised. This assumption shaped everything: surgical protocols, drug development, our entire understanding of infection and immunity. And it was based on the best evidence available at the time. When doctors took samples from these tissues and tried to culture bacteria in labs, nothing grew. The petri dishes stayed clean. The conclusion seemed obvious, but the conclusion was wrong. The problem wasn't the tissues, it was the method. Most bacteria can't be cultured in standard lab conditions. They require specific temperatures, specific nutrients, specific chemical environments, specific partnerships with other organisms. Growing them in isolation on agar plates is like asking a fish to survive on dry land. It doesn't work. The absence of growth doesn't mean absence of life. It means we were looking in the wrong way. Then came DNA sequencing, and everything changed. Starting in the 2010s, researchers began extracting DNA directly from supposedly sterile tissues, blood, bladder, lungs, placenta, and sequencing it. And they found bacterial DNA, not contamination, not experimental error, actual bacterial genomes present in tissues we thought were lifeless. The bladder, for instance. For decades, urinary tract infections were diagnosed based on culture results. If bacteria grew from a urine sample, you had an infection. If nothing grew, you were healthy. Simple. But research published in 2023 by Alan Wolf's lab at Loyola University revealed that the bladder hosts a resident microbiome. Healthy bladders contain bacteria, Lactobacillus, Gardnerella, Streptococcus, and others, at low but consistent levels. They're not causing disease, they're just there, living in the urine, in the bladder lining, possibly providing benefits we're only beginning to understand. Women with overactive bladder or urinary incontinence have different bladder microbiomes than healthy women. The differences are subtle, shifts in species composition rather than presence versus absence, but they're consistent. And they suggest that bladder health isn't just about keeping bacteria out, it's about maintaining the right bacterial community. So now we're rethinking urinary tract infections. Maybe some UTIs aren't invasions by external pathogens. Maybe they're ecological collapses, where resident species that normally keep the system stable are displaced by opportunistic species that trigger inflammation. And the bladder isn't unique. The lungs were also thought to be sterile, except during active infections. You breathe in bacteria constantly. Every breath carries microbes from your mouth, your nose, the surrounding air. But the assumption was that healthy lungs clear them immediately. Cilia sweep them up, mucus traps them, immune cells engulf them. Within hours, the lungs return to sterile baseline, except they don't. According to research from 2024 by Leopoldo Siegel at NYU Grossman School of Medicine, healthy lungs contain bacterial communities. They're sparse compared to the gut, maybe 1,000 bacteria per milliliter of lung fluid compared to billions per milliliter in the colon, but they're there. Species from the mouth and throat constantly migrate into the lungs, and some establish residence. Prevotella, Veillonella, Streptococcus, these are common residents. Their populations stay low, controlled by immune surveillance, by the mechanical clearing mechanisms of the respiratory system, by competition with each other. But they persist, and their presence matters. People with chronic obstructive pulmonary disease, or COPD, have different lung microbiomes than healthy people. Asthma patients have different profiles. Cystic fibrosis patients have dramatically altered communities, often dominated by Pseudomonas aeruginosa, a species that forms biofilms in the thick mucus characteristic of the disease. Whether these microbial changes cause the diseases or result from them is still being debated, but evidence suggests it's bidirectional. Disease alters the lung environment, which favours certain bacterial species, which produce metabolites and trigger immune responses that worsen the disease. It's a feedback loop, And if lungs have microbiomes, what about the heart? The brain? Blood was the ultimate sterile frontier. The cardiovascular system is sealed, pressurized, constantly monitored by immune cells. Any bacteria detected in blood is treated as sepsis, a life-threatening emergency requiring immediate antibiotic intervention. But studies using sensitive DNA sequencing methods have found bacterial DNA in blood samples from healthy people, not at levels suggesting active infection, just traces, fragments, entire genomes sometimes, suggesting that intact bacteria are circulating at very low levels, evading immune detection, possibly even living inside white blood cells. This is controversial. Many researchers argue that the DNA is from dead bacteria, debris being cleared from the gut or other tissues, not evidence of living populations. Others point to studies showing that bacteria can indeed survive in blood, that certain species have evolved mechanisms to resist immune attack, to hide inside cells, to persist in a dormant state until conditions improve. The implications are staggering. If blood isn't sterile, if bacteria are constantly circulating at low levels, then maybe chronic diseases we don't understand, atherosclerosis, some autoimmune conditions, even aspects of aging, involve microbial components we've been missing because we weren't looking for them. And then there's the brain. The brain has always been considered immune privileged, protected by the blood-brain barrier, a tight seal of endothelial cells that prevents most molecules, most pathogens, most immune cells from entering neural tissue. The barrier keeps the brain sterile, safe, isolated from the microbial chaos of the rest of the body. But research published in 2024 by Courtney Walker at the University of Alabama found bacterial DNA in post-mortem brain tissue from people who died of non-infectious causes. Proteobacteria, firmicutes, actinobacteria, species typically found in the gut or mouth, somehow present in brain tissue. The findings are preliminary, controversial. The researchers are careful to note that they can't prove the bacteria were alive, can't confirm they weren't post-mortem contamination, can't say whether they were causing damage or existing benignly. But if validated, the implications reshape neuroscience. Could gut bacteria, under certain conditions, cross the blood-brain barrier? Could they contribute to neuroinflammation, to conditions like Alzheimer's disease, where chronic inflammation seems to precede and possibly trigger neurodegeneration? There's precedent. Certain pathogens are known to invade the brain. Borrelia burgdorferi, the bacterium that causes Lyme disease, can cross the blood-brain barrier and cause neurological symptoms. Treponema pallidum, the syphilis bacterium, does the same. So the barrier isn't impenetrable. It's a filter, not a wall. And if pathogenic bacteria can cross it, maybe commensal species can too under the right or wrong conditions. This is speculative territory, the edge of current knowledge. But it reveals how much we still don't know about the invasions that shape us, about the boundaries between self and microbial other, about whether any part of the human body is truly, permanently sterile. But internal tissues aren't the only frontier. There's another invasion happening, one that's even more intimate, that begins before you're born and continues every night while you sleep. Let's talk about the places you rest. Your bed is an ecosystem. Right now, folded into the fabric of your sheets, embedded in the fibers of your mattress, living in the pillows you press your face against every night, are organisms feeding on you. Not attacking you, not biting or burrowing, just grazing. Consuming the constant rain of biological debris you shed without noticing. You produce this debris continuously. Dead skin cells, as we've mentioned, roughly 30,000 to 40,000 per hour. Oils from sebaceous glands. Sweat, even when you don't feel like you're sweating, evaporating from your skin at rates of about half a litre per night. Moisture from your breath. Heat from your metabolism. All of it accumulates in your bed. And where there's organic matter, there's life. Dust mites are the most abundant. Dermatophagoids terranicinus and Dermatophagoids farini, two species that have adapted specifically to human environments that can't survive outdoors, that depend entirely on the controlled temperature and humidity of human homes and the constant supply of shed skin cells. They're tiny, about 1/4 millimetre long, just barely visible to the naked eye if you know what you're looking for. 8 legs, oval bodies, translucent exoskeletons. Under a microscope, they look like bloated ticks, which makes sense. They're arachnids, relatives of spiders and scorpions, Though they don't bite and don't have venom, they just eat, constantly, grazing on skin flakes on the fungi that also grow on skin debris, on bacteria, on any organic particle small enough to consume. And they're everywhere. A typical mattress after a few years of use can contain millions of dust mites. Millions! The weight of the mattress increases measurably over time, not from your body but from the accumulated biomass of mites, their feces, their shed exoskeletons, their dead bodies decomposing into dust. And that dust is a problem. Not the mites themselves, they're harmless. But their feces contain proteins, digestive enzymes that trigger allergic reactions in sensitive individuals. These proteins become airborne when you move in bed, when you pull back covers, when you walk across carpet. You breathe them in, your immune system recognizes them as foreign, and in some people, it overreacts. According to research from the Asthma and Allergy Foundation of America, roughly 20 million Americans are allergic to dust mites. The symptoms mimic seasonal allergies: sneezing, runny nose, itchy eyes, congestion. But it's not seasonal, it's year-round, constant. Because the mites are always there, and they're not easy to eliminate. Washing bedding in hot water kills them, but they recolonize within days. Reducing humidity helps. Mites need moisture to survive, but most homes maintain humidity levels comfortable for humans, which are also comfortable for mites. Encasing mattresses and pillows in allergen-proof covers prevents mite feces from becoming airborne, but the mites themselves continue living inside. You can't sterilize your bed, you can only manage the population, keep it from exploding, maintain it at levels your immune system can tolerate without chronic inflammation. But dust mites aren't alone in your bed. There are also fungi, Aspergillus, Penicillium, Cladosporium species growing in the warm, moist environment created by your body. They feed on the same organic debris as dust mites, competing with them, sometimes parasitizing them, forming food webs that operate on scales of millimeters. And there are bacteria. Staphylococcus species transferred from your skin, Corynebacterium species thriving in the oils you shed, environmental species brought in from outside, hitchhiking on clothes, on pets, on air currents flowing through open windows. Your bed is a microcosm, a miniature ecosystem with producers, consumers, decomposers, predators, prey. Energy flows through it. Nutrients cycle. Populations rise and fall based on temperature, humidity, how often you wash your sheets, how much you sweat, whether you sleep alone or with a partner or a pet. And it's not unique to beds. Every surface in your home is colonized. Your furniture, your carpets, your walls. The air you breathe contains bacterial cells, fungal spores, pollen, dust mite feces, all of it suspended in invisible clouds that settle slowly, that get stirred up with every movement, that you inhale with every breath. Indoor air quality researchers estimate that a typical home contains about 9,000 different species of fungi and bacteria, not individuals, species. The diversity is comparable to outdoor environments, though the composition is different. Indoor microbiomes are shaped by the people who live there, by their skin microbiomes, by their activities, by how they clean, how they cook, whether they have pets, how much ventilation they allow. According to research from 2024 by Noah Fearer's lab at the University of Colorado, you can identify who lives in a house based on the microbial communities on surfaces. Each person has a unique microbial signature, a distinct pattern of species they shed more than others that colonize the spaces they occupy most frequently. The bathroom reflects the people who use it. The kitchen reflects the food they prepare. The bedroom reflects who sleeps there. And these communities aren't static. They change seasonally, with humidity and temperature. They change when you clean, when you introduce new furniture, when you open windows. They respond to antibiotics you take, to illnesses you have, to stress and diet, and whether you're exercising regularly. Your home's microbiome is an extension of your body's microbiome. The two are in constant exchange, constantly influencing each other, creating feedback loops that shape both. But homes are relatively recent inventions. For most of human evolution, we lived outdoors, in caves, in temporary shelters. We were exposed to environmental microbes constantly, directly, without the filtering effects of walls and roofs and HVAC systems, and that exposure shaped us. Our immune systems evolved in a world teeming with microbial diversity. Soil bacteria, plant-associated bacteria, bacteria from animals, from water, from rotting vegetation. We encountered thousands of species daily, and our immune systems learned to navigate that complexity, to distinguish between harmless environmental species and actual threats. Modern environments are different, cleaner, more controlled, and while that's reduced infectious disease dramatically, saved millions of lives, improved health in countless ways, it's also created a new problem. We're not encountering the diversity our immune systems expect. The hygiene hypothesis, first proposed in 1989 by epidemiologist David Strachan, suggests that reduced microbial exposure in childhood contributes to rising rates of allergic and autoimmune diseases. The immune system, deprived of the training it evolved to receive, becomes dysregulated. It attacks harmless substances. It attacks the body's own tissues. It makes mistakes, and evidence supports this. Children raised on farms have lower rates of asthma and allergies than urban children. Children with pets have more diverse home microbiomes and lower allergy rates. Children delivered vaginally, exposed to maternal microbes during birth, have different immune development than children born via C-section. Exposure matters. Diversity matters. The invasions we experience in early life, the microbial communities we encounter and integrate into our bodies, shape our health for decades to come. But not all invasions happen early. Some are opportunistic. Some wait for moments of weakness. There are organisms that wait, dormant, silent, encoded in your genome, woven into the fabric of your DNA, patient as stone. They're not bacteria. They're not fungi. They're something older, something stranger. They're viruses, or what's left of viruses. Ghosts of ancient infections that happened so long ago, the hosts weren't even human yet. Roughly 8% of your genome is viral, not genes you inherited from your parents in the usual sense, not mutations that accumulated over generations. These are sequences inserted by retroviruses, viruses that reproduce by integrating their genetic material directly into host chromosomes. When they infect a cell, they hijack its machinery, copy their RNA genome into DNA, and splice that DNA into the host's genome, permanently. Most of the time, this happens in somatic cells, skin, liver, muscle, and when the cell dies, the viral sequence dies with it. But occasionally, rarely, a retrovirus infects a germ cell, a sperm, an egg. And when that cell participates in fertilization, the viral DNA gets passed to the next generation. It becomes heritable, part of the species. This has happened thousands of times over the course of mammalian evolution. Retroviruses infected our ancestors, integrated into their genomes, and got locked in. Most of these sequences, called endogenous retroviruses or ERVs, are now defunct, mutated beyond recognition, broken remnants incapable of producing functional viruses, but they're still there, scattered throughout your chromosomes like fossils in sedimentary rock, evidence of infections that happened millions of years ago, and some of them are still active. Not as infectious agents, as regulatory elements, switches that control gene expression, that turn other genes on or off, that shape how your cells develop and function. According to research published in 2024 by Cedric Fechot at Cornell University, endogenous retroviruses play essential roles in placental development. The placenta, that temporary organ that connects mother and fetus, that allows nutrient exchange without mixing blood directly, relies on proteins originally encoded by ancient retroviruses. Syncytin is one. It's a fusogenic protein, meaning it causes cells to fuse together, creating the multinucleated structure of the placental barrier. Without it, the placenta doesn't form properly. Pregnancy fails, and syncytin isn't a human invention. It's a viral protein, hijacked from a retrovirus that infected a mammalian ancestor roughly 25 million years ago, repurposed, integrated into the reproductive machinery of an entire lineage. We didn't evolve pregnancy. Viruses did. We just borrowed their tools. And syncytin isn't unique. There are dozens, possibly hundreds, of endogenous retroviral sequences that have been co-opted, domesticated, turned into functional genes. Some are involved in immune regulation, some in brain development, some in stem cell pluripotency, the ability of embryonic cells to differentiate into any tissue type. So when you think about invasion, about colonization, about organisms living inside you, don't just think about bacteria and fungi. Think about the viral DNA woven into every cell in your body, sequences that were once invaders that are now part of what makes you human. But endogenous retroviruses are ancient history, fossilized, the invasions that happened before you were born. There are also current invasions, viruses circulating in your body right now, and most of the time you don't notice them. Herpes viruses are the perfect example. There are 8 human herpes viruses, and most adults carry at least 3. Herpes simplex virus type 1, the cause of cold sores, infects roughly 67% of people worldwide. Varicella zoster virus, which causes chickenpox in childhood, persists lifelong and can reactivate decades later as shingles. Epstein-Barr virus, the cause of infectious mononucleosis, infects more than 90% of adults globally, and once you're infected for life. Herpes viruses are masters of persistence. After the initial infection, after your immune system mounts a response and clears the active virus from your bloodstream, some viral particles retreat. They migrate into neurons, into sensory ganglia, clusters of nerve cells near your spinal cord, and there they go dormant. They stop reproducing, stop producing proteins, they become latent, invisible to the immune system, undetectable by any diagnostic test that relies on detecting active viral replication. They're just sitting there, waiting, and occasionally, unpredictably, they reactivate. Stress, illness, immune suppression, UV exposure, something triggers the dormant virus to wake up. It starts replicating, producing new viral particles, traveling back down the nerve to the skin or mucous membranes, causing lesions, sores, rashes. Your immune system responds again. The outbreak is controlled, the virus retreats back into dormancy, and the cycle continues, potentially for your entire life. This is not an infection in the way we usually think of infections. It's not something you catch and then clear. It's a negotiated truce, a balance between viral replication and immune suppression maintained at the level of individual neurons, with neither side winning completely. And herpes viruses aren't unusual. Many viral infections follow similar patterns. Cytomegalovirus, another herpes virus, establishes lifelong latency in most people, occasionally reactivating, occasionally transmitting to new hosts through saliva or other bodily fluids. Human papillomavirus persists in epithelial cells for years, sometimes decades, occasionally causing warts or, in certain strains, contributing to cancers. The line between infection and colonization blurs. These viruses aren't separate from you. They're part of your biological reality, shaping your immune function, influencing your disease susceptibility, even affecting your behavior in ways we're only beginning to understand. Research from 2023 by Robert Yolkin at Johns Hopkins University found correlations between latent viral infections and neuropsychiatric conditions. People with schizophrenia have higher rates of Toxoplasma gondii antibodies, suggesting prior infection by this brain-dwelling parasite. People with bipolar disorder show elevated antibodies to certain herpes viruses. The mechanisms are unclear, but the patterns are consistent enough to suggest that chronic latent infections might influence brain chemistry, neuroinflammation, even cognition. This is controversial territory. Correlation isn't causation. But the hypothesis is testable, and studies are underway exploring whether antiviral treatments or immune modulation could affect psychiatric symptoms. If true, it would mean that mental health isn't purely neurological or purely psychological. It's also microbial, another layer of complexity in the already bewildering ecosystem of the human body. But viruses, for all their strangeness, are at least familiar. We know they exist. We test for them. We have vaccines and treatments. There are other invaders we've only recently begun to acknowledge. Ones that don't fit into any existing category. Prions, for instance. These aren't alive. They're not organisms. They're proteins. Misfolded proteins that can propagate their misfolded shape, converting normal proteins into copies of themselves, spreading through tissue like a chain reaction. Prions cause diseases like Creutzfeldt-Jakob disease in humans, scrapie in sheep, chronic wasting disease in deer and elk. The symptoms are neurological, dementia, loss of coordination, personality changes. The diseases are always fatal, and there's no cure, no treatment, no way to stop the spread once it starts. But prions are rare. Most people will never encounter them. There are other, more common entities that occupy the space between living and non-living that challenge our definitions, that force us to rethink what invasion even means. Extracellular vesicles, for example. These are tiny membrane-bound packets, roughly 30 to 1,000 nanometers in diameter, shed by nearly every cell type in your body. They contain proteins, RNA, sometimes DNA, packaged and released into extracellular fluid, into blood, into spaces between tissues. For decades, they were considered cellular garbage, waste products, cells shedding debris to clear out damaged components. But research over the past 15 years has revealed something astonishing. These vesicles are communication devices. Cells package specific molecules into vesicles and send them to other cells. The vesicles travel through the bloodstream, cross tissue boundaries, bind to target cells and deliver their contents. The receiving cell incorporates the molecules and its behavior changes. Gene expression shifts. Signaling pathways activate. The cell responds as if it received a message. And it did. Vesicles are letters, telegrams, packets of information moving through the body, carrying instructions, warnings, updates from one tissue to another. But here's where it gets strange. Vesicles aren't just moving between your own cells, they're moving between you and your microbes. Bacteria produce extracellular vesicles. Gut bacteria package proteins and RNA into vesicles and release them into the intestinal lumen. Some of these vesicles cross the intestinal barrier, enter the bloodstream, travel to distant organs. They've been found in the liver, in the brain, in the placenta. According to research published in 2024 by Sarkis Mazmanian's lab at Caltech, bacterial vesicles from the gut can cross the blood-brain barrier and influence neuronal activity. They deliver bacterial metabolites, small molecules that affect neurotransmitter production, that modulate inflammation, that alter synaptic function. So your gut bacteria aren't just living passively in your intestines, they're communicating with your brain, sending signals, influencing how neurons fire, how circuits form, how you think and feel. This is the gut-brain axis made tangible. Not a metaphor, not a vague connection, a physical communication system operating through vesicles, through molecules, through chemistry that crosses barriers and links ecosystems. And it's bidirectional. Your cells send vesicles to your bacteria. Your immune cells package antimicrobial peptides into vesicles and release them into the gut, targeting specific bacterial species, shaping the community composition. Your intestinal epithelial cells send vesicles containing RNA, and bacteria take up that RNA, and it affects their gene expression, their metabolism, their behavior. You're not separate from your microbes. You're in constant conversation with them, negotiating, coordinating, influencing each other in ways that blur the boundaries between host and symbiont, between self and other. And this conversation isn't limited to bacteria. Your cells communicate with fungi, with viruses, with every organism that colonizes you. The invasion isn't one way, it's reciprocal. It's a dialogue that's been running for millions of years, that started long before humans existed, that will continue long after we're gone. But for all this talk of microscopic invaders, of organisms too small to see, there's one invasion we haven't discussed yet. One that's visible, tangible, one that people notice, that they react to with visceral disgust, that shapes behavior and culture and hygiene practices in ways microscopic organisms never could. Let's talk about the insects you can see, the ones that bite. There are insects that have been hunting humans for so long they can't survive without us. The human body louse, Pediculus humanus, is one. It's a tiny insect, roughly 3 millimeters long, wingless, flattened, with claws perfectly adapted for gripping fabric fibers. It lives in clothing, in the seams of shirts and pants, emerging several times a day to feed on blood from the skin beneath. And it's entirely dependent on humans. Body lice can't survive on any other species. They can't tolerate the lower body temperature of other mammals. They can't navigate fur the way head lice can navigate hair. They require the specific temperature range maintained by clothed human skin, the specific chemical environment of human sweat, the specific texture of woven fabric. This dependency is so absolute that body lice can be used as evolutionary markers. By sequencing their DNA and comparing genetic differences between populations, scientists can trace human migration patterns, estimate when clothing was invented, reconstruct the history of human civilization through the parasites we carried with us. According to research published in 2023 by David Reed at the University of Florida, body lice diverged from head lice roughly 170,000 years ago. This means humans were wearing clothing by then, creating the ecological niche that allowed a new louse species to evolve. Before clothing, there were only head lice. After clothing, the population split. Some lice stayed on the scalp, others colonized the new habitat humans had created, fabric. So clothing didn't just protect us from the elements, it created invasions, it enabled new parasites, it changed the selective pressures on both humans and the organisms living on us. And body lice aren't just annoying, they're vectors, they carry diseases. Epidemic typhus, caused by the bacterium Rickettsia prowazekii, is transmitted by body lice. The bacteria live in the lice's gut, and when a louse feeds, it defecates. The feces contain bacteria, and when you scratch the bite, you rub the bacteria into the wound. Infection follows. Epidemic typhus has killed millions throughout history. It spreads in conditions of overcrowding, poor sanitation, inability to wash clothing regularly, war, famine, displacement. Situations that force people into close quarters without adequate hygiene infrastructure. The disease-shaped military campaigns influenced the outcomes of battles, collapsed armies not through combat but through infections spread by lice living in soldiers' uniforms. Napoleon's retreat from Russia in 1812 is estimated to have killed more soldiers through typhus than through freezing or combat. The trenches of World War I were breeding grounds for lice, for typhus, for misery that went beyond the violence of warfare itself. And even now, in refugee camps, in prisons, in any situation where people are crowded together without access to clean clothing or adequate bathing facilities, body lice persist. Typhus re-emerges, the invasion continues, opportunistic, waiting for conditions that favour its spread. But body lice at least stay in clothing. They don't burrow into skin. They feed and retreat. There are other insects that go deeper. Scabies mites, Sarcoptes scabiei, are parasites that tunnel. They're arachnids, relatives of spiders and ticks, roughly 0.4 millimeters long, just barely visible as tiny moving dots on skin if you look closely. But most of the time, you can't see them. They're under the surface. Female scabies mites burrow into the outermost layer of skin, the stratum corneum creating tunnels as they move. They lay eggs in these tunnels, 2 to 3 eggs per day, and the eggs hatch within days. The larvae emerge, mature, mate on the skin surface, and the cycle repeats. The mites themselves aren't particularly harmful, but your immune system reacts to them, to their saliva, to their feces, to the proteins they shed as they tunnel. And the reaction is intense, itching, rashes, inflammation that worsens over weeks as your immune system becomes sensitized, as the allergic response builds. Scratching damages the skin, creates openings for bacterial infections. Secondary infections with Staphylococcus or Streptococcus species are common, sometimes severe, occasionally leading to sepsis or kidney damage from immune complexes. Scabies is highly contagious, spreading through prolonged skin-to-skin contact. It doesn't jump or fly. The mites crawl slowly from one person to another. So transmission requires closeness, sleeping in the same bed, caring for children, sexual contact, any situation where bodies touch for extended periods. And once established, scabies is difficult to eliminate. The mites can survive for several days away from human skin, living in bedding, in clothing, in furniture. Treating the person isn't enough. You have to treat the environment, wash everything in hot water, vacuum thoroughly, isolate contaminated items for at least a week, long enough for any surviving mites to starve. According to the World Health Organization, scabies affects roughly 200 million people globally at any given time. It's most common in tropical regions in areas with limited access to clean water and healthcare, but it occurs everywhere. Outbreaks happen in nursing homes, in schools, in hospitals. Anywhere people are in close contact, scabies can spread. And there's a more severe form. Crusted scabies, sometimes called Norwegian scabies, occurs when the immune system fails to control mite populations. Instead of a few dozen mites, the infected person harbours thousands, sometimes millions. The skin becomes thick, crusted, covered in scales that contain massive numbers of mites and eggs. This happens most often in people with weakened immune systems, HIV patients, organ transplant recipients, elderly people with dementia who can't maintain hygiene. The condition is extremely contagious, far more so than typical scabies, because the number of mites shedding from the skin is exponentially higher. Treatment is challenging. Standard anti-scabies medications often fail because the thick crusts prevent the drugs from penetrating. Aggressive therapy is required, sometimes involving systemic medications, repeated applications, hospitalization to manage secondary infections. But scabies mites, for all their invasiveness, are at least treatable. There are other insects that invade human tissue in ways that are harder to control, harder even to detect. Myiasis is the infestation of living tissue by fly larvae. Maggots. Most people associate maggots with decomposition, with dead tissue, with corpses. But some fly species lay eggs on living hosts. The larvae hatch, burrow into skin, and feed on living tissue. Botflies are the most infamous. There are several species, each adapted to different hosts: cattle, horses, rodents. But some, like Dermatobia hominis, the human botfly, target humans. The adult fly doesn't bite or sting. Instead, it captures a mosquito or another biting insect, glues its eggs to the insect's body and releases it. When the mosquito feeds on a human, the body heat triggers the botfly eggs to hatch. The larvae drop onto the skin and burrow into the bite wound. Once inside, the larva creates a chamber, a space just beneath the skin where it lives and grows. It breathes through a small opening, a pore connected to the surface, and over the course of several weeks it feeds, molts, develops, eventually reaching a size of up to 2 centimeters. The host, the human, feels it. A lump under the skin, movement sometimes, pain, itching, a sense that something is wrong. But identifying the problem isn't always straightforward. The symptoms can resemble a cyst, an abscess, an infection. Removal requires care. If you squeeze too hard, the larva can rupture, releasing its contents into the surrounding tissue, triggering a severe inflammatory response. If you try to pull it out with tweezers and it breaks, the remaining parts continue to decay inside, causing infection. The traditional method, still used in regions where botflies are common, involves covering the breathing pore with petroleum jelly or tape, suffocating the larva, forcing it to back out toward the surface where it can be grasped and extracted. It's effective but slow, sometimes taking hours. Surgical removal is faster, but requires anesthesia, an incision, careful extraction to avoid leaving fragments behind. And botflies aren't the only species that infest human tissue. Screwworm flies, Cochleomia hominivirax, lay eggs in open wounds. The larvae hatch and burrow into living flesh, feeding voraciously, expanding the wound, creating cavities that can extend deep into muscle, even into bone. Before its eradication from North America in the 1960s through a massive sterilization campaign, screwworm infestations killed thousands of livestock annually and occasionally infested humans, particularly in rural areas where injuries went untreated. The larvae would consume tissue faster than it could heal, creating horrific wounds, secondary infections, sometimes death from sepsis. The species still exists in parts of Central and South America, and occasional reintroductions into North America require immediate response to prevent re-establishment. The border between the United States and Mexico is monitored constantly, with sterile flies released as a preventive measure, millions of them, rendering wild populations unable to reproduce. This is one of the most successful examples of biological control in history. An invasion stopped not by killing the invader, but by disrupting its reproduction, by flooding the environment with sterile males that outcompete fertile ones, driving the population into collapse. But not all insect invasions can be controlled so elegantly. Some are too widespread, too adaptable, too deeply integrated into human environments. Bed bugs, Cimex lectularius, are resurging. For decades, from the 1950s through the 1990s, they were rare in developed countries. DDT and other broad-spectrum insecticides had nearly eliminated them. But restrictions on pesticide use, combined with increased international travel, created opportunities for bedbugs to return. And return they did. Starting in the late 1990s, bedbug infestations began appearing in hotels, apartments, dormitories, homes. By the 2010s, they were epidemic in some cities: New York, London, Paris, Sydney. No region was immune. Bedbugs are nocturnal feeders. They hide during the day in cracks, crevices behind baseboards, inside mattress seams. At night, attracted by carbon dioxide and body heat, they emerge. They walk across sheets, climb onto skin, pierce with needle-like mouthparts, and feed for 5 to 10 minutes. Then they retreat. The bites aren't immediately painful. Bedbugs inject an anaesthetic along with anticoagulants, numbing the area, preventing blood from clotting. You don't feel it happening. You wake up with welts, with itching, with lines of bites across exposed skin where the bug moved and fed multiple times, and the psychological impact is severe. Knowing that insects are crawling on you while you sleep, feeding on you, that your bed, the place you're supposed to feel safest, is infested, creates anxiety, insomnia, obsessive behaviours. People throw out furniture, move apartments, develop phobias. Eliminating bedbugs is expensive and difficult. They're resistant to many common insecticides. They can survive months without feeding, waiting in dormancy for a host to return. They spread through walls, through ventilation systems, from one apartment to another, turning isolated infestations into building-wide crises. Heat treatment is effective. Raising the temperature of an entire room to 120 degrees Fahrenheit for several hours kills all life stages. But it requires specialised equipment, professional application, and it's costly. Freezing works too, but only at extreme temperatures, below zero for extended periods, which is impractical for most living spaces. So bedbugs persist, they adapt, they spread, and they remind us that even in modern sanitised environments, even with all our technology and chemistry and knowledge, we can't fully control the invasions that target us. But bedbugs, for all their resurgence, are at least confined to buildings. They don't follow us outside. There are other insects that hunt us wherever we go, that have shaped human history, human settlement patterns, human health on scales most people don't realise. Mosquitoes have killed more humans than any other animal in history, not through venom, not through direct tissue damage, through the diseases they carry: malaria, dengue, yellow fever, Zika, West Nile virus, chikungunya, lymphatic filariasis. The list is staggering. Hundreds of millions of infections annually, hundreds of thousands of deaths. And that's now, with modern medicine, with insecticides, with anti-malarial drugs and mosquito nets and public health infrastructure. Go back a century and the toll was far worse. Malaria alone may have killed half of all humans who ever lived. That's not hyperbole. That's demographic modelling based on the evolutionary pressure malaria has exerted on human genetics, on the frequency of protective mutations like sickle cell trait, on the distribution of blood type variants that confer resistance. Malaria shaped us. It killed so many people, so consistently for so long, that natural selection favoured any genetic variation that provided even slight protection, even if that variation had costs. Sickle cell disease, for instance, is devastating when you inherit 2 copies of the mutated gene, but 1 copy provides resistance to severe malaria. So in regions where malaria is endemic, where the risk of infection is high, carrying 1 sickle cell allele is advantageous. It increases your chances of surviving to reproduce. The disease shaped the genome. The parasite, through the mosquito, through infection after infection across millennia, changed what it means to be human in malaria zones. This is evolution in action. Selection pressure so strong it alters allele frequencies in populations within dozens of generations, and it's still happening. Malaria hasn't been eliminated, it's been pushed back, confined mostly to sub-Saharan Africa, parts of South Asia, scattered regions in South America. But 247 million cases were reported in 2021, according to the World Health Organization. 619,000 deaths, mostly children under 5. The parasite, Plasmodium falciparum being the deadliest of 5 species that infect humans, has a complex life cycle. It reproduces sexually in the mosquito gut, producing sporozoites that migrate to the salivary glands. When the mosquito bites, sporozoites are injected into the bloodstream. They travel to the liver, invade hepatocytes, multiply for a week or 2 without causing symptoms. Then they burst out. Thousands of merozoites from each infected liver cell flooding into the blood, invading red blood cells. Inside the red blood cells, they multiply again. The cells rupture. more merozoites are released, more red blood cells are invaded. The cycle repeats every 48 to 72 hours depending on the species, and with each cycle symptoms spike: fever, chills, sweating. The classic periodic pattern of malaria. Your immune system detects the parasite, releases cytokines, triggers inflammation, but the parasite is always one step ahead, hiding inside cells, changing surface proteins to evade antibodies, surviving just long enough to reproduce and move to new cells before the old ones are destroyed. Some merozoites differentiate into gametocytes, sexual forms that don't cause symptoms but circulate in the blood, waiting. If a mosquito feeds, if it takes up blood containing gametocytes, the cycle continues. The gametocytes fuse in the mosquito gut, produce more sporozoites, infect the next person the mosquito bites. It's elegant in a terrible way. The parasite has two hosts, both required, both exploited. Humans provide the environment for asexual reproduction, for amplification, for producing the biomass needed to infect mosquitoes. Mosquitoes provide the environment for sexual reproduction, for genetic recombination, for creating the diversity that allows the parasite to evade drugs and immune responses. And the mosquito, Anopheles species primarily, is perfectly adapted for its role. Only females feed on blood. They need the protein for egg development. Males feed on nectar. So malaria transmission is driven entirely by female mosquitoes seeking nutrients to reproduce. They hunt by detecting carbon dioxide, the gas you exhale with every breath. From dozens of meters away, they sense the plume, follow the concentration gradient, home in on the source. Closer in, they detect body heat, lactic acid from your skin, other volatile compounds that signal a warm-blooded host. They land, probe with their mouthparts, piercing skin, seeking a capillary. When they find one, they inject saliva containing anticoagulants, the same compounds that cause the itching and swelling of mosquito bites. And if the mosquito's infected, sporozoites are injected along with the saliva. One bite. That's all it takes. One infected mosquito, one moment of exposure, and the parasite is in you. But malaria isn't the only threat mosquitoes carry. Dengue fever infects roughly 390 million people annually, according to 2024 estimates from the World Health Organization. The virus, transmitted primarily by Aedes aegypti mosquitoes, causes high fever, severe joint and muscle pain. Dengue used to be called breakbone fever because of how intense the pain is, and occasionally progresses to hemorrhagic fever, a life-threatening condition involving internal bleeding, shock, organ failure. There's no specific treatment, no antiviral drug, just supportive care, fluids, rest, hoping the immune system can clear the virus before complications develop. And dengue has a unique feature: antibody-dependent enhancement. If you're infected once, you develop immunity to that strain. But there are 4 different dengue serotypes, and immunity to one doesn't protect against the others. Worse, if you're infected with a second serotype, antibodies from the first infection can actually help the virus infect cells more efficiently, enhancing the disease severity rather than preventing it. So your second dengue infection is often worse than your first, your third worse than your second. Each infection primes your immune system to make the next one more dangerous. It's a paradox, a failure of the immune memory that usually protects us, turned into a vulnerability. Yellow fever, another mosquito-borne virus, killed thousands during the construction of the Panama Canal. Workers died faster than they could be replaced. The project stalled. It wasn't until public health efforts targeted mosquito breeding sites, drained standing water, introduced screens and nets, that the death rate dropped enough for construction to continue. The disease shaped infrastructure. It determined where cities could be built, which trade routes were viable, which regions could be colonized. Mosquitoes, through the diseases they carried, were geopolitical forces, and they still are. Zika virus emerged as a major threat in 2015, spreading rapidly through the Americas, causing microcephaly in infants born to infected mothers. The virus crosses the placental barrier, infects fetal neural tissue, disrupts brain development. Children are born with abnormally small heads, severe cognitive impairments, lifelong disabilities. The outbreak reshaped public health policy, sparked urgent research into mosquito control, into vaccines, into understanding how viruses interact with developing nervous systems. And then, as quickly as it surged, Zika receded. not because it was eliminated, because enough people were infected that herd immunity developed, reducing transmission rates below the threshold needed to sustain an epidemic. But the virus is still circulating, still present in mosquito populations, still capable of resurging if conditions change, if immunity wanes, if the virus evolves to evade existing antibodies. Mosquitoes are vectors, but they're also ecosystems. They carry parasites and viruses, yes, but they also carry bacteria. Wolbachia, a genus of bacteria that infects many insect species, lives inside mosquito cells, particularly in reproductive tissues. It's passed from mother to offspring through eggs, and it manipulates mosquito reproduction to favour infected females. In some cases, Wolbachia makes male mosquitoes sterile. In others, it causes eggs fertilised by uninfected males to die. The result is that Wolbachia spreads rapidly through mosquito populations, even though it provides no direct benefit to the mosquitoes themselves. But here's where it gets interesting. Wolbachia also interferes with virus replication. Mosquitoes infected with Wolbachia are less capable of transmitting dengue, Zika, chikungunya. The bacteria compete with the viruses for resources, trigger immune responses that suppress viral loads, make the mosquito a less effective vector. This has been weaponized. Starting in 2011, researchers began releasing Wolbachia-infected mosquitoes into the wild, particularly Aedes aegypti populations in dengue endemic regions. The bacteria spread through the populations naturally, driven by the reproductive manipulation mechanisms, and within a few years most mosquitoes in the release areas were infected, and dengue transmission dropped. Not to zero, but significantly. Studies from Indonesia, Brazil and Australia have shown 70 to 90% reductions in dengue cases in areas where Wolbachia-infected mosquitoes were released. This is biological control, but subtler than the sterile male technique used against screwworms. Instead of eliminating the vector, you're modifying it, making it less capable of transmitting disease while allowing it to persist in the ecosystem. It's a compromise, a negotiation. And so far it's working. But mosquitoes aren't the only insects that bite, that feed on blood, that transmit diseases in the process. Ticks are arachnids, 8-legged relatives of spiders, and they're vectors for a staggering array of pathogens. Lyme disease, transmitted by Ixodes scapularis, the black-legged tick, is the most well-known in North America. The bacterium Borrelia burgdorferi causes a characteristic bullseye rash in the early stages, followed by fever, fatigue, joint pain. If untreated, it can spread to the nervous system, the heart, causing chronic symptoms that persist for years. Rocky Mountain spotted fever, transmitted by dermacentaur ticks, is caused by Rickettsia rickettsii, a bacterium that invades endothelial cells lining blood vessels. The infection causes vasculitis, inflammation of blood vessels, leading to rashes, fever, and in severe cases, organ failure and death. It's one of the deadliest tick-borne diseases in the Western Hemisphere, with case fatality rates of 20 to 30% if untreated. And ticks are expanding their ranges. Climate change is allowing species that were once confined to southern regions to move northward, bringing diseases with them. Lone star ticks, Amblyomma americanum, are spreading from the southeastern United States into New England, the Midwest, even parts of Canada. They transmit ehrlichiosis, a bacterial infection similar to Rocky Mountain spotted fever, and they're also associated with alpha-gal syndrome, a bizarre allergic reaction to red meat. Alpha-gal syndrome develops after repeated bites from lone star ticks. The tick's saliva contains a sugar molecule called galactose alpha 1-3-galactose, or alpha-gall, which is also present in mammalian meat, beef, pork, lamb, but not in primates or birds. When the tick bites, it introduces alpha-gal into your bloodstream. Your immune system recognizes it as foreign and produces antibodies. Later, when you eat red meat, the alpha-gal in the meat triggers an allergic reaction. Not immediately, like most food allergies, but hours later, after the meat has been digested and the alpha-gal molecules are absorbed into the bloodstream. The delay makes diagnosis difficult. People don't connect the reaction to the meal they ate hours earlier. Symptoms range from hives and gastrointestinal distress to anaphylaxis, a life-threatening reaction involving swelling, difficulty breathing, shock, and there's no cure. The only treatment is avoiding red meat entirely. The allergy can fade over time if you're not bitten again, but repeated bites reinforce the sensitization. making the allergy permanent. So a tick bite can change your diet, change what foods you can tolerate, change your relationship with a fundamental category of nutrition. It's an invasion that alters behavior, culture, social interactions, all through a molecule injected by a tiny arachnid feeding for a few days on your blood. But ticks and mosquitoes, for all the diseases they carry, are at least temporary. They bite and leave. There are insects that invade more permanently, that colonize the body, that establish residence. Tungiasis is an infestation caused by Tunga penetrans, the Shigo flea. The female flea burrows into skin, usually on the feet, embedding herself in the tissue. Her abdomen swells as eggs develop, expanding to the size of a pea, distorting the surrounding skin. She remains there for weeks, releasing eggs through a small opening, feeding continuously on blood from the host. The infestation is painful. The swollen flea creates pressure, distorts the foot structure, makes walking difficult. Secondary bacterial infections are common, sometimes leading to tetanus, gangrene, amputation in severe cases. Removal requires careful extraction, cutting away the flea and surrounding tissue without rupturing the abdomen, which would release eggs and inflammatory fluids. In regions where tungiasis is endemic, parts of sub-Saharan Africa, the Caribbean, Central and South America, the infestation is a chronic public health problem, particularly in communities with poor access to footwear, where people walk barefoot on contaminated soil. These aren't just medical issues, they're social issues, economic issues. Children with tungiasis miss school, adults miss work. The cycle of poverty and disease reinforces itself, with infestations contributing to reduced productivity, reduced opportunity, reduced quality of life, and addressing it requires more than medicine. It requires shoes, clean floors, education about hygiene, infrastructure to reduce contact between bare feet and flea-infested environments. The invasion is biological, but the solutions are social. But not all invasions are hostile. Some are invited, some are cultivated, some we depend on more than we realize. Consider your eyelashes. Right now, at the base of nearly every follicle on your eyelids, there are mites. Demodex follicularum and Demodex brevis, the same species we discussed earlier. But here on the eyelashes they serve a function we're only beginning to understand. They eat debris, dead skin cells, excess oils, bacteria that would otherwise accumulate and clog the follicles. In normal numbers, they're maintenance workers, janitors keeping the ecosystem balanced. And your immune system tolerates them, has learned over your lifetime that these mites are part of the baseline, part of the normal state of your body. But tolerance requires calibration. If mite populations grow too large, if the balance tips, inflammation follows. Blepharitis, chronic eyelid inflammation, is often linked to Demodex overgrowth. The eyelids become red, swollen, itchy. The follicles crust over with debris and mite waste. Vision can be affected if the inflammation spreads to the cornea. Treatment involves reducing mite populations, tea tree oil, specific lid scrubs, sometimes ivermectin, but also supporting the conditions that keep populations stable. Proper eyelid hygiene, managing underlying conditions like rosacea or seborrheic dermatitis that create environments where mites thrive. It's not about eliminating the mites, it's about restoring the balance, accepting that these organisms are residents, not invaders, and that health comes from coexistence, not eradication. This is true across your entire body. The organisms living on you, in you, aren't aberrations. They're features, integral components of a functioning system. And when we try to eliminate them completely, when we sterilize too aggressively, we often create more problems than we solve. Antibacterial soaps are a perfect example. For decades, they were marketed as superior to regular soap, capable of killing 99.9% of bacteria, making your hands cleaner, safer, and they did kill bacteria. But they killed beneficial bacteria along with harmful ones. They disrupted skin microbiomes, creating opportunities for opportunistic species to colonize, sometimes species more problematic than the ones that were eliminated. According to research published in 2024 by the FDA, antibacterial soaps containing triclosan are no more effective at preventing illness than regular soap and water. The mechanical action of washing, scrubbing, rinsing removes bacteria just as effectively without the chemical disruption. And regular soap doesn't select for antibiotic resistance the way antibacterial agents do. Triclosan has since been banned in consumer hand soaps in the United States, but it took decades of use, decades of environmental accumulation, decades of potential harm to microbiomes and ecosystems before the evidence was clear enough to act. The lesson is broader than triclosan. It's about recognizing that sterility isn't health, that microbial diversity is protective, that the goal isn't to eliminate organisms but to maintain communities that function, that resist pathogens, that support rather than undermine physiological processes. This is a fundamental shift in medical thinking. For most of the 20th century, the germ theory paradigm dominated. Microbes cause disease. Eliminate microbes, eliminate disease. It worked brilliantly for acute infections, for conditions caused by single pathogens like tuberculosis or cholera or syphilis. Antibiotics saved millions of lives. Vaccines eradicated smallpox, nearly eradicated polio. But the paradigm struggles with chronic conditions, with diseases that involve microbial imbalances rather than invasions. Inflammatory bowel disease, obesity, metabolic syndrome, allergies, autoimmune disorders. These aren't caused by single pathogens. They're linked to disrupted microbiomes, to communities that have lost diversity, lost key functional species, lost the resilience needed to maintain homeostasis, and treating them requires different strategies, not killing, restoring, not antibiotics, probiotics, prebiotics, dietary interventions that feed beneficial species, fecal transplants that reintroduce entire communities. We're moving from a war model to an ecological model, from fighting invasions to managing ecosystems. And the shift has profound implications. It means recognizing that your health isn't just about your human cells. It's about the trillions of non-human cells living on and in you, their metabolic outputs, their interactions with your immune system, their influence on everything from digestion to mood to cognition. It means understanding that antibiotics, for all their power, have costs. Every course of antibiotics disrupts your microbiome, kills beneficial species, selects for resistant ones. Sometimes that cost is necessary. The infection you're treating is life-threatening, and the microbiome can recover. But often, antibiotics are prescribed unnecessarily. For viral infections they can't treat. For mild bacterial infections the immune system could clear on its own, and the cumulative damage adds up. Research from 2024 by Martin Blaser at Rutgers University estimates that the average child in the United States receives 10 to 20 courses of antibiotics before adulthood. Each course reduces microbial diversity, alters community composition, potentially creates vulnerabilities that persist for years. And this is happening at a population level. We're conducting an unintentional experiment, exposing entire generations to repeated microbial disruptions, and we're only beginning to measure the consequences. Rising rates of autoimmune diseases, allergies, metabolic disorders. These correlate with antibiotic use, with reduced microbial exposure, with changes in how we live that have insulated us from the microbial diversity our immune systems evolved to expect. This isn't an argument against antibiotics. They're essential, life-saving. But it's an argument for using them judiciously, for preserving microbial diversity as a health priority, for recognizing that not all bacteria are enemies. Some are allies, some are neutral, and some occupy categories we don't have good words for. Organisms that are harmful under certain conditions but beneficial under others, whose effects depend entirely on context, on what other species are present, on the state of your immune system, on factors we're still identifying. Helicobacter pylori is one, Staphylococcus epidermidis is another. It lives on everyone's skin, usually harmlessly, sometimes beneficially, by producing antimicrobial compounds that inhibit more dangerous species. But it can also cause infections, particularly if it enters the bloodstream through a wound or surgical incision, forming biofilms on medical devices like catheters and prosthetic joints. Is it a pathogen or a commensal? Both. Neither. It's a species occupying ecological niches, behaving according to the opportunities and constraints it encounters. Context determines outcome, and humans are part of that context. Our behavior, what we eat, how we clean, what medications we take, how much we sleep, how stressed we are, shapes the microbial communities living on us. We're not passive hosts, we're active participants in the ecosystems we carry. When you eat fibre, you're feeding gut bacteria that produce short-chain fatty acids. When you exercise, you alter the composition of your gut microbiome, increasing species associated with improved metabolism and reduced inflammation. When you're stressed, cortisol and other stress hormones affect gut permeability, microbial gene expression, immune function. The invasions aren't separate from your life. They're woven into it, responding to it, shaped by it. And this creates opportunities. If your behavior influences your microbiome, and your microbiome influences your health, then you have leverage. Not complete control. Genetics and early life exposures set baselines that are difficult to change, but meaningful influence. Dietary interventions are the most studied. Increasing fibre intake promotes beneficial gut bacteria. Fermented foods, yogurt, kefir, kimchi, sauerkraut, introduce live bacteria that can temporarily colonize the gut or support resident species. Polyphenols from fruits, vegetables, tea, coffee act as prebiotics, feeding specific bacterial species that produce anti-inflammatory compounds. According to research from 2024 by Tim Spector's group at King's College London, people who eat more than 30 different plant species per week have more diverse gut microbiomes than people who eat fewer than 10. The diversity of your diet predicts the diversity of your microbiome, and microbial diversity correlates with metabolic health, immune function, even mental health outcomes, so the invasion can be shaped, curated. You can't choose which species colonize you. Many are acquired in early childhood, established before you had any agency. But you can influence which species thrive, which ones dominate, which ones are kept in check by competitors. This is empowering, but it's also humbling, because it means you're not in complete control. You're a participant in a negotiation that's been ongoing for millions of years, a conversation between your cells and microbial cells, between your immune system and their evolutionary strategies. And the negotiation continues, constantly, in every tissue, every surface, every interface between your body and the world. There's a place where the invasion becomes most visible, most tangible, where the boundary between inside and outside collapses completely. Your mouth, again. But this time, not the bacteria, the architecture they create. Dental plaque, that biofilm we mentioned earlier, isn't just a collection of bacteria, it's a structure, a city. With layers, neighbourhoods, infrastructure connecting different zones, waste management systems, communication networks. It's one of the most complex bacterial communities accessible to study without invasive procedures, and it's taught us more about how microbes organise themselves than almost any other system. The biofilm begins with pioneer species, Streptococcus mutans, Streptococcus sanguinis, species that can attach directly to clean tooth enamel using adhesins, proteins that bind to calcium and proteins in your saliva. They arrive within minutes of brushing, floating in saliva, landing on teeth, testing the surface, attaching if conditions are right. Once attached, they start dividing. They produce extracellular matrix, polysaccharides and proteins that form a sticky scaffold around them, and they release signaling molecules, chemical broadcasts that say, "We're here, the surface is good, come join us." Other species respond. They can't attach to clean enamel on their own, but they can attach to the pioneer species, binding to proteins on their cell surfaces, building upward, outward, creating layers. Fusobacterium nucleatum is a common secondary colonizer, a bridge species that many other bacteria can attach to, linking early and late colonizers, facilitating the development of mature biofilms. According to research published in 2023 by Jessica Mark Welch at the Marine Biological Laboratory, dental plaque develops in predictable patterns. The same species show up in the same spatial arrangements, forming characteristic structures that are consistent across individuals. It's not random, it's organized. Near the base, close to the tooth, are aerobic species, bacteria that need oxygen. At the surface of the biofilm, exposed to saliva and air, oxygen is abundant. But as the biofilm thickens, as layers build up, oxygen becomes scarce. The middle and deep layers become anaerobic, and different species dominate. Bacteria that can't tolerate oxygen, that produce different metabolic byproducts, that fill different functional roles. This vertical stratification creates chemical gradients. Oxygen decreases with depth, pH varies, nutrient availability shifts, and bacteria position themselves according to their metabolic needs, their tolerances, their competitive abilities. But they're not just stacking randomly, they're forming specific structures. Corncob formations, where a central filamentous bacterium is surrounded by smaller cocci attached in radial patterns. Hedgehog structures, dense clusters with projections extending outward. These aren't accidents. They're adaptive architectures, shapes that maximize surface area, that facilitate nutrient flow, that protect interior bacteria from immune cells and antimicrobial agents. And the biofilm communicates internally. Bacteria produce and detect quorum-sensing molecules, coordinating gene expression across the community. When populations are high, when the biofilm is mature, certain genes activate. Genes for producing more matrix, genes for antibiotic resistance, genes for producing toxins or enzymes that break down host tissue. The biofilm becomes more than the sum of its parts. It's a multicellular organism in its own right, with specialization, division of labor, coordinated responses to environmental challenges. And it's incredibly difficult to remove. Brushing disrupts the surface, breaks off chunks, but deeper layers remain. Flossing removes biofilm from between teeth, but only the areas you reach. Within hours, the biofilm begins rebuilding. Within days, it's back to baseline. Antibiotics don't penetrate well. The matrix blocks many drugs, and even if the drug reaches interior bacteria, they're often in dormant states, not actively dividing, which makes them resistant to antibiotics that target cell division or protein synthesis. This is why chronic infections associated with biofilms, dental infections, but also infections on prosthetic joints, catheters, heart valves, are so hard to treat. The bacteria aren't just individual cells floating freely. They're organized, protected, coordinated. Attacking them requires disrupting the entire structure, not just killing individual cells. And this organizational capacity isn't unique to dental plaque. Biofilms form on every surface in your body where bacteria have time to attach and accumulate. Your gut lining has a mucus layer colonized by biofilms. Your skin has localized biofilms in hair follicles, in sweat glands. Medical implants, pacemakers, artificial joints, catheters develop biofilms within days of insertion. The invasion creates architecture, and that architecture changes the rules, makes bacteria harder to eliminate, harder to control, harder to understand using models based on free-floating cells in test tubes. Microbiology for decades studied bacteria as isolated species in pure cultures, grow them in broth, on agar, measure growth rates, test antibiotic susceptibility. It was clean, reproducible, convenient. But it was also misleading, because bacteria in nature, bacteria in real environments, including the human body, don't live in pure cultures. They live in communities, mixed species, interacting constantly. And their behavior in communities is radically different from their behavior in isolation. Species that seem harmless alone become dangerous in combination. Species that appear pathogenic in culture turn out to be beneficial in context. The properties of the community can't be predicted from the properties of individual species. This is emergence, complexity arising from interaction. And it means that understanding the invasion shaping your body requires studying them as ecosystems, not as collections of individual organisms, which brings us back to the question we started with. Where do you end and the invasion begins? Your skin is covered in organisms. Your gut is filled with them. They're in your mouth, your lungs, possibly your blood. They're living on your furniture, in your bed, in the air you breathe. They're communicating with your cells, influencing your immune system, affecting your brain, and you're influencing them. What you eat, how you live, the choices you make, all of it shapes the communities that colonize you. So the boundary blurs. You're not a discrete organism, you're a meta-organism, a holobiont, a collective entity composed of human and microbial cells functioning together. And this has implications that extend beyond medicine, beyond biology, into philosophy, into how we think about identity, autonomy, individuality. If your microbiome affects your mood, your behavior, your decisions, then are those decisions entirely yours? If bacterial metabolites cross the blood-brain barrier and alter neurotransmitter levels, influencing how you feel and think, then where does your agency end and microbial influence begin? These aren't just abstract questions, they're practical. If we develop treatments that modify the microbiome to alter behaviour, using probiotics to treat depression, for instance, or fecal transplants to improve cognitive function, are we treating the person or manipulating their symbionts? And if the person and the symbionts are inseparable, does the distinction even matter? We've built our entire medical system, our legal system, our sense of self, on the assumption that humans are individual organisms, discrete, autonomous, responsible for their actions. But that assumption is increasingly hard to defend. You're not one genome, you're thousands. You're not one metabolism, you're an integrated network of metabolisms, human and microbial, producing compounds neither could make alone, responding to stimuli in ways that depend on the composition of your microbial communities. This isn't reductionism. It's not saying you're nothing but bacteria. You're obviously more than that. You have a brain, consciousness, subjective experience, agency, but you have those things in conversation with the organisms living in and on you, not in isolation from them. And recognizing that changes everything. It changes how we treat disease. It changes how we think about health. It changes what it means to be human. Because if humans have always been holobionts, if every person who ever lived was a walking ecosystem, then the invasions we've been describing aren't invasions at all. They're relationships, partnerships, sometimes symbiotic, sometimes parasitic, often somewhere in between. And the goal isn't to repel them, it's to understand them, to manage them, to cultivate the relationships that support health and limit the ones that cause harm. This is ecological medicine. Precision medicine, yes, but precision applied to entire communities, not just individual species or individual human genes. We're at the beginning of this shift. Most medical interventions still target single organisms or single pathways. Antibiotics kill bacteria. Antivirals inhibit viral replication. We're good at removing threats, less good at restoring balance. But the field is changing. Microbiome research is exploding. Thousands of studies published annually. Clinical trials testing probiotic interventions, fecal transplants, dietary modifications. Companies developing live biotherapeutics, engineered bacteria designed to colonize the gut and produce therapeutic compounds. Some of this will fail. Many probiotics don't colonize effectively. Many dietary interventions show inconsistent results. The microbiome is complex, variable, resistant to simple manipulation, but some will succeed. We're learning which interventions work for whom, under what conditions. We're developing tools to measure microbiome composition more accurately, more cheaply, more quickly. We're building models that predict how communities will respond to perturbations, and eventually we'll reach a point where managing your microbiome is as routine as managing your diet or exercise, where doctors prescribe microbial interventions alongside pharmaceutical ones, where maintaining microbial diversity is recognized as a public health priority, like vaccination or clean water. We're not there yet. But the trajectory is clear. The invasions aren't going away. They can't. They're part of what we are. But we can shape them, guide them, work with them instead of against them. And that might be the most profound realization. The organisms living on you aren't enemies. They're not even really invaders. They're companions, partners in a relationship that began millions of years ago and continues in every breath, every bite, every moment of your life.